Hello, this is my lab discussion video. I'm doing the digital enzyme catalysis lab from, I think it was a week ago. But yeah, let's get right into this. Okay, so the introduction, basically the main idea for this lab, the main question they were asking is what is the effect of temperature on the activity of the catalase enzyme. It's pretty much this lab was just testing different temperature ranges and to see the effectiveness of the enzyme at each temperature range. Explored the optimal temperature range for the enzyme based on what they gave in the lab. Pretty sure it was 5 degrees, 25 degrees, and 75 degrees. Correct me if I'm wrong. My hypothesis, of course, was 25 degrees would be the most optimal temperature for this specific enzyme. I based this basically not necessarily common sense, but regular cells can't operate when it's too hot or too cold, so I figured 25 degrees is in the middle, so it would be the best option for this lab. The data, there were two forms of data collected. There was a table full of the values. I believe it was seconds, and then the amount of oxygen that was released over time. And then I condensed that into a bar graph of my own. Just through going through the lab, it gives you the numbers, and then I was just able to chart that and then plot it on the graph. So just recorded that, and I'm going to show you what I found. Let's go ahead. Here's our data. This is the data table. I was correct. So it was time within 0 to 180 seconds, and then the amount of oxygen that was produced at 5 degrees, 25 degrees, and 75 degrees. As you can see here, 25 degrees proved to be the most effective, had the most released. 75 degrees had the least amount, and then 5 degrees was right in the middle. Then over here is the graph. Maybe a little hard to see. You might want to zoom in, but pretty much just corresponding. This is the O2 numbers. This is the seconds that goes by. This first one corresponds with 5 degrees. The middle one corresponds with 25 degrees. And then the third one, of course, corresponds 75 degrees. And then I just plotted that along the axes, increasing as time goes on. Well, not very much for this one, but increasing nonetheless. For my results, as we could see in this graph in the table, the optimal temperature was around 25 degrees, which is 77 degrees Fahrenheit. From this, we can pretty much figure that the optimal temperature range for catalase is at least 25 degrees, maybe a little higher, maybe a little lower, but it at least includes that value. I was actually a little surprised just how little was found in the 5 degrees, because that's really, I think that's 5 degrees Celsius is around 40 Fahrenheit, so not super cold, but I figured there would be a bigger cutoff for that because, not too hot, not too cold. 40 degrees just seems a little high for it to be that low. As for my conclusions, as I previously said, the optimal temperature was 25 degrees. Basically, the main idea from this is that catalase performs best at a warmer temperature, not too hot, not anywhere near the hundreds. I'd say, for my assumptions, going off of 25 degrees, probably around 80 to 95, if I had to guess, somewhere in that range. It supports my hypothesis because now we know that the optimal range includes at least 25 degrees Celsius. The lab provides valuable information because we could basically just use this as a baseline. We already know that enzymes, they can become desaturated or denatured by temperature and other factors. So just by going off of this, we know that in the catalase example, the effective range is 25 degrees. So we could just base that. You could set up a similar experiment with other enzymes, do the same procedures, and then you could find out what the effective range is for a different enzyme. So I think it helps to kind of solidify the fact that temperatures can affect enzymes and how productive they are, how much they can do, which is how productive they are. Yeah. That's pretty much about it. Thank you for watching.